So as we've already seen in our video about the central dogma, protein synthesis is a two-step process. And the first step of that process is transcription, where we go from DNA to RNA, and that is what this video is going to be about. There are three main steps to transcription in general, which are initiation, elongation, and termination. And I'll tell you where these steps are in the steps that I've listed here. Initiation, I'm just going to put a big I next to this. Initiation starts when an RNA polymerase enzyme binds to the DNA molecule. Okay? And so RNA polymerase sounds exactly like a different enzyme we were just studying called DNA polymerase, and it's sort of similar. It's an enzyme, and it will make a polymer of RNA. Okay? And so after it binds to the sort of starting section of a DNA molecule, the DNA molecule will unzip, and a new RNA strand is assembled by adding RNA nucleotides in a complementary manner. Okay? And so this is basically when you start to have elongation. And so the big thing here to note is that actually during DNA replication we had both helicase and DNA polymerase. Here we just have RNA polymerase. RNA polymerase does the job of both helicase. It opens the DNA double helix, and the polymerase enzyme, because it not only opens, it also adds new nucleotides at the same time. The other thing that you should be noticing here in this picture is that unlike DNA replication, we are only opening the DNA up for a short period of time. Notice that RNA polymerase is moving this way. And then, look at this, the DNA closes back up again. So in transcription, we're only reading a single gene and then closing sort of the book of DNA. It's like we're looking at a page or two and making a copy, closing it again. So that's initiation and elongation, and this is now termination. Transcription ends when the RNA polymerase reaches what is considered a termination signal on the DNA molecule, okay? And then the RNA molecule, which is shown here in red, is then released and will ultimately exit the nucleus. This is just a different picture of transcription. I very frequently like to show students multiple pictures of things just so that, on a test or something like that, you might come across a different picture and you should be able to read that. Notice that we're sort of looking at DNA vertically here. Notice that RNA polymerase is this sort of gray blob of protein sitting here, and transcription is obviously proceeding in this direction because the already written mRNA molecule is sort of extending out this way.